Good morning. This is Subra Suresh, President of Nanyang Technological University in Singapore. It's a great pleasure and privilege to introduce our plenary speaker this morning, Professor Robert Langer from the Massachusetts Institute of Technology. Professor Langer is a longtime friend and colleague. Let me take a few minutes to give an introduction. Professor Langer is one of 11 institute professors at the Massachusetts Institute of Technology. He has written over 1,500 articles, which have been cited over 320,000 times. He has more than 1,400 issued and pending patents worldwide. His patents have been licensed or sub-licensed to over 400 companies. He has served as chairman of the Federal Drug Administration in the US Science Board, its highest advisory board, from 1999 to 2002. His awards, which are over 220, are too numerous to list here. I will just mention a few. He has received both the US National Medal of Science and the National Medal of Technology and Innovation, the Charles Stark Draper Prize, which is the Engineering Nobel Prize, equivalent to the Engineering Nobel Prize given by the National Academy of Engineering in Washington, D.C., the Queen Elizabeth Prize for Engineering, the Breakthrough Prize in Life Sciences, the Kyoto Prize, the Millennium Technology Prize, and many, many others. The Lemelson MIT Prize recognized him for being one of history's most prolific inventors in medicine. He holds 34 honorary doctorate degrees, and he is one of very few Americans elected to all three branches of the US National Academies: the National Academy of Engineering, the National Academy of Sciences, and the National Academy of Medicine. In addition to that, he has been elected to the American Academy of Arts and Sciences and the National Academy of Inventors. So it's my great pleasure to welcome my good friend, Professor Robert Langer. The title of the plenary talk is The Edison of Medicine: Robert Langer's Quest to Solve Global Health Challenges Using Biotechnology. Bob.
Thank you. Thanks so much. Thank you.
Well, thank you very much. It's a pleasure to be able to speak with you. I'd like to start by telling you a little bit about my career when I was the age of many of you. When I was like a lot about your old age, I was a graduate student at MIT in chemical engineering. And when I got done with my doctoral degree, pretty much all my friends, this was 1974, went into work for oil companies. That's what chemical engineers did. So I thought that's what I should do too, just what my friends did. So in all the oil companies, there was like a gas shortage around then, and they had all these high-paying jobs. So I applied to the oil companies, and I actually got 20 job offers, 4 actually from Exxon alone. And I wasn't that good, but they just had so many openings. One of them made quite an impression on me. I remember going to Exxon in Texas or Louisiana, and when I went there, one of the older engineers said to me, if you could just increase the yield of this one chemical by 0.1%, he said, that would be wonderful. He said it'd be worth billions of dollars. And I remember flying home that night thinking to myself that I really didn't want to do that. And you know, I just thought it would be boring and unimportant. So it made me think about what I did want to do. And when I was a graduate student at MIT, in addition to doing my thesis, I helped start a school for poor high school children. And I got very involved in it, and I helped develop new chemistry curriculum, new math curriculum, to try to make math and science very interesting for people even who had dropped out of school. And so one day I saw an ad to be assistant professor at what's called City College of New York. And it was to do just that, develop new exciting curriculum. So I wrote them a letter applying for a job as an assistant professor, but they didn't write me back. But I really liked that idea, so I found all the ads I could to do something like that. I found about 40, and I wrote to all those colleges, and actually not a single one wrote me back. So that wasn't going so well. So I started to think, what other way could I use my chemical engineering education to help people? So I thought about medicine. So I applied to a lot of hospitals and medical schools. They didn't write me back either. But one day, one of the people in the lab where I was, one of the older people, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But anyhow, I wrote to Dr. Folkman, and he was kind enough to offer me a job as a postdoc at Boston Children's Hospital and Harvard Medical School. And the problem that he wanted me to work on is shown on the next slide. He had this idea that if you could stop blood vessels, maybe you could stop cancer. And if you look at this picture, this was from 1971, actually. On the bottom left, you see a depiction of a tumor cell. Somehow it's become abnormal and it starts growing abnormally. And then as you see it, as you see the curve going, it grows bigger. It grows to about, if you look in the middle, the lower center of the screen, what you see is it's probably about 1 million cells. And it won't actually get bigger than that because it runs into a nutrition problem, except for one thing. He says that the tumor, he believes, creates a chemical signal called TAF, or tumor angiogenesis factor. And what that does is it causes the surrounding blood vessels, which normally don't do anything, to start growing right to the tumor. And they connect with the tumor, and that solves the nutrition problem for the tumor. And then, as you see on the upper right of this picture, the tumor gets bigger and bigger because it has blood vessels in it. And if you go to the uppermost picture, you see that the tumor is highly vascularized, and actually it spreads through those blood vessels. That's a process called metastasis, which often kills people. So my job, he said, was to prove that he was right. It turned out that this was an incredibly controversial theory. Almost everybody at this time in the early 70s didn't believe it. They said it wasn't right. And he wanted me to prove it, but in so doing, isolate the first substances that could stop blood vessels from growing. So how do you think about a problem like that? Well, we broke it down to 2 parts. First, where could you ever find something that would stop blood vessels? And we thought about cartilage, which is in your nose and your knee, and it doesn't have blood vessels. So I was able to get like little rabbits in the lab, but I couldn't get much cartilage from them. So in chemical engineering, you learn about scale up. So I went to cows and I got some cow bones at a local slaughterhouse, but still getting 1 or 2 bones, you can't get that much from that either. So I tried to find out, where do all the cow bones in the northeastern United States go? And it turns out they go to these meat packing places in South Boston. So I made an arrangement with them to get all their bones. I'd go there 4 times a week. I'd bring their bones back. It totally occupied my entire car. And I'd bring them back, and I'd scrape the meat off of the bones. Let me just show you a picture on the next slide. And this is a cow bone, and the top of it's cartilage. You see when the meat's scraped off, it's like white and glistening, and there are no blood vessels. So what I do is I slice off that cartilage piece in the top. I'd extract it in solvents like with guanidinium hydrochloride to break the non-covalent bonds, and then I'd put it through different purification procedures. So that at the end of about a couple of years, I had like what I'll call 100 different fractions from the cartilage, and I needed to test them. So that brings up the second problem: how do you test whether you're stopping blood vessels from growing? And what you need to do, and this almost always has been an issue in any type of time in history when anybody's ever found a new factor, is you have to have what's called a bioassay. And there was no bioassay for studying blood vessels, so we had to invent one. And what Dr. Folkman and I tried to do is shown on the next slide. This was our idea, that maybe you could put a certain type of tumor, we call a V2 carcinoma, in the eye of a rabbit. Rabbits have big eyes, so they're easy to visualize through an ophthalmic microscope. And also the tumor will bring blood vessels in from the edge of the cornea, what's called the limbus, but it takes about 9 weeks. So what you want is something we thought that could be not inflammatory in the eyes, like maybe a polymer or lipid, something like that, so it wouldn't cause any damage. And then it would also have to be able to release these molecules in the eye for whatever period of time we wanted, like say 9 weeks in this case. And the beauty of the eye is that now you could actually use an ophthalmic microscope to measure the length of the longest blood vessels. So Dr. Folkman, he was on the board of the only company in the world that had ever tried to deliver it's called ALSA. They had Nobel laureates in chemistry on their board, and it was a very distinguished group. And he asked them, could they help us? But what they said, next slide, is that you couldn't do it. They basically said, if we go to this next slide, that you can't do that because we wanted to use polymers, and they said the big molecules, they can't get through them. It's like any of us walking through a wall. I mean, you might be able to make it very, very porous, but then it comes out immediately. So the challenge is, could we slowly deliver these molecules for at least a long enough time to do something? In fact, the literature said the same thing. Next slide. And really, I think the only thing I had going for me is I hadn't read any of this. So I tried anyhow. And I spent 2 years working in the laboratory, experimenting with many different techniques, different materials, different designs. And I found over 200 different ways to get this to not work. But eventually, I found out a way to make little tiny particles, microspheres, nanospheres. And let me just show you a picture on the next slide. Here you see on the left is a picture of one of these, and on the right, it's cut in half. And what we did, next slide, is we published in the journal Nature in 1976 that you could use this to release anything, any protein. And actually, this was also, it turns out, and this will come into my talk later, it's actually the very first time anybody developed a delivery system for nucleic acids like RNA or DNA. So, so that was very exciting to me that we could deliver all these molecules. In fact, we even, as we went further, worked out ways to release them constantly if we wanted to. And a picture of that is shown in the next slide. So I was all very excited about that. But then in 1976, around the time of that paper, I was asked to give a big speech at a major scientific conference in Michigan. And I was 27. I was the youngest person there. And I practiced this talk for weeks. And finally, I got up and and there were all these very distinguished scientists at the meeting and who were experts in materials and engineering and chemistry. I got up and I gave that talk. It was a 20-minute talk. And at the end of it, I actually thought I did all right. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk, all these older scientists, being nice people, would want to encourage me, this young guy. But when I got done, I stepped off the podium and a whole bunch of them came up to me and they said, "We don't believe anything you just said." They said, next slide, that the large molecules are just too big to get through. And we were also using organic solvents. And they said that will denature whatever you put in, peptides, proteins, nucleic acids. So they said, this has got to be wrong. And actually, because of all that, things went downhill from there. Actually, I tried to get research grants. My first 9 grants were rejected. And then a lot of my friends said I should try to get a job, not just be a postdoc. So, I listened to them, and I'm a chemical engineer, like I said, so I applied for a lot of faculty positions in chemical engineering departments. But not a single chemical engineering department in the world would hire me. So I didn't get any jobs there. Finally, Dr. Folkman, he knew a man named Nevin Scrimshaw, who was head of the nutrition department at MIT, and he introduced me to him. And Dr. Scrimshaw was kind enough to offer me a job. But the only thing is, Dr. Scrimshaw was a great man, but he was what I'd almost call like a benevolent dictator. He liked me, so he hired me, but he didn't ask anybody else in the department what they thought. That probably would have been okay, except for one thing. And that is, the year after I joined the department, Dr. Scrimshaw left. So a lot of the senior faculty decided to give me advice. And their advice is I should leave too. In fact, on the next slide, I tell you a story. This is from a few years ago, actually. Michael Marletta, he's a very famous chemist. He was head of Berkeley's chemistry department, head of Scripps, member of the U.S. National Academy of Sciences. He was giving a speech when I got an award, and he was saying what it was like to be with me in the late 1970s and early 80s at MIT, and I'll just read this to you. He said one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said a senior scientist sat quizzing us while smoking a cigar. He said when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, "You better start looking for another job." Mike said he thought he was in a Fellini movie. So that was very discouraging, but I didn't have much choice. I just kept trying to work, even though nobody believed it. I tried to understand it better. And I had one of my graduate students, Rajan Bawa, cut thin slices through our systems using a cryomicrotome, a low temperature cutting instrument. So on the next slide, let me just show you a picture of one of those. And so if molecules were really big, they couldn't get through this. Now, it turns out when we put like a model molecule in, like we picked the red protein on the next slide, and then we put it in using the way we made these, it's looked very different. This is a thin section, a 5-micron thin section cut before any release has taken place. And you see what we call a phase separation. Polymer is the white part, and the drug, the myoglobin, so to speak, that's the red part. Now, if you release this, say, for a year, and then you look at it, it looks tremendously different. And that's shown on the next slide. So here, what you see are all these pores that have been formed. They weren't there 2 slides ago. And it turns out that these pores are interconnected. They're also incredibly winding and tortuous. So it takes a very, very long time for the molecules to get through. And they're also, even though they're connected, the pores, there are tight constrictions between them. And so that makes a really long time for the molecule to get through. And it turns out over the years, a lot of my students, like Bill Ryan and Dean Shea and Larry Brown, Mark Saltzman, they've worked out mathematical models to predict exactly how you can control these pore structures, what materials you can use, so you can get almost any release rates you want from days to years or any time in between. Well, anyhow, since we had laid this foundation, now we could go back and solve the angiogenesis problem. So the next slide shows the assay that I mentioned. And I apologize in advance for the slide I'm going to show you now because it's a little bit graphic. It has blood vessels in it. But there's no other way to illustrate this. So what we're going to do is look at this 9 weeks later. We actually did 2,000 eyes, 100 fractions or so. And I'm only going to show you representative ones from the paper. But this is at 9 weeks. If we could just go to the next slide, what you'll see is on the left-hand side without the cartilage-derived inhibitor. And this is true for almost every fraction we tested, too, because almost every one didn't work. But what you see on the left is the sheet of blood vessels growing over the polymer to the tumor. And in fact, if you looked at this eye 3 weeks later, it would be three-dimensional. Out of the orbit of the eye, it would kill. But when we put the inhibitor in, or the one thing that turned out to be the inhibitor in, take a look on the right-hand side. And we repeated this 21 times. And you see the vessels are lower, they're sparser, and they actually avoid the polymer. And 60% of the time, these tumors never grew. So that was very exciting, and that showed for the first time that blood vessel inhibitors did exist. And also it provided a way for future people to study this. Now, this is 1976 academic study. It still took another 28 years from our paper and billions of dollars and great companies like Genentech doing work on this before the first blood vessel inhibitor was approved by FDA. And let me just show you what happened starting 28 years later. Next slide. What you see is starting in 2004, 28 years after we started, is one after the next, after the next, after the next blood vessel inhibitor approved by the FDA. Avastin, the first of these, is which was originally approved for colorectal cancer, is probably the #2 largest selling biotech drug in history. And of course, these have been approved for many, many different diseases, many types of cancer. And not only that, they've been widely used. In fact, they're the only therapy, pharmacologic therapy for diseases of blindness in the back of the eye, like macular degeneration and diabetic retinopathy. In fact, sometimes they can actually reverse these diseases. So those very historic, and these are used in tens of millions of people every year now. So that showed that you could actually, and now many of these, and this continues to today, have these approvals by FDA. And these drugs are all blockbusters, sell many billions a year. But, I also thought that maybe the drug delivery work we've done could be useful in its own right. So one of my graduate students at MIT, Larry Brown, just as a proof of principle, showed, as we'll see on the next slide, that you could design, and I'm just showing you one graph from his whole thesis, but if we go to the next slide, what you could see is he was able to lower the blood sugar level of a diabetic rat from 400, which is diabetic, to 100, which is normal, for over 100 days. And yet that's with a single implant. And so one day, Dr. Folkman and Dr. Folkman said to me, Bob, we should file a patent. And so we did. But 5 years in a row, the patent examiner rejected it. In fact, it's interesting. This was the first patent in the history of Boston Children's Hospital, which is a very good hospital. But we've got rejected 5 years in a row. So the head of the technology transfer office at the hospital came to see me one day. He said, Bob, you have to stop. He said, this examiner is never going to allow this patent. Just forget it. You're wasting an enormous amount of money for the hospital. He said, just give up. But I don't like to give up. So I started to think, how could we convince the patent examiner to allow this patent, legally, of course? And science really wasn't working. But as I told you before, when I first started doing this, everybody told me it was impossible. It could never work. So I wondered, did anybody ever write that down? So you can do what's called a science citation search. I did this in 1982. So remember, the Nature paper we wrote was 1976. So I looked back, see who cited that paper and what they said. And I found hundreds of references. But I thought I'd just show you one. And this is on the next slide. So this has actually turned out to be written in 1979. I had no idea. By the way, it's an interesting exercise to see who writes about what you do. I had never done this before. So, but this was really very helpful. These 5 people wrote that describing the field that the agent to be released is a small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, they said, like proteins, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising. So surprising is a really good word to show to a patent examiner. Surprising results that clearly demonstrate the opposite. So I showed that to the head of the tech transfer office. He showed it to the patent examiner. And the patent examiner said, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from these 5 scientists that they really wrote that. So I was this young guy, but I assistant professor. I wrote them all, and they're all nice enough to write me back that they really did write that. And the patent examiner allowed the patent. The next slide shows that. With that, we would start different companies. We would teach different people how to do this, teach different companies how to make these. And what's happened because of that, next slide, is there are many, many products used really probably by hundreds of millions of people all over the world. These are just a few, but there are, like in the middle right of the screen, there's Lupron Depot. That's a little microsphere. If you tried to swallow, luteinizing hormone, releasing hormone analogs, it won't work because they'll get destroyed and they're too big to be absorbed. So they tried to inject it, but they were destroyed that way too. So they put them in these little microspheres, inject them subcutaneously, and they become a widely used therapy for hundreds of millions of patients. And there are many, many others. New ways of treating opioid addiction, pain, cancer, heart disease, different eye diseases, ways to stop bleeding, and mental health diseases like schizophrenia. All these are different ones that have been approved here, type 2 diabetes. So that was very rewarding to me to see that could happen. Also, one of the things we did is not only, a lot of these that I'm showing you are microspheres that you might inject intramuscularly or subcutaneously, But we also thought we could make nanospheres and inject them into the body to do different things. And as I mentioned to you, the Nature paper was the first time anybody put in large molecules and those as well. So I thought I'd show you, and we also figured out ways. One of the other problems is when you inject nanospheres into the body, they get destroyed right away. And we developed certain types of coating, like what's called polyethylene glycol. It attracts a lot of water. And that coating actually enables the nanospheres to last long enough to do good. And the next slide is going to show you a video that explains this far better than me. I wish I could do as well.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
Now, I should point out that a lot of the clinicians I work with tell me it doesn't blow the cell up quite like that. But this kind of nano-type approach works on all kinds of things. We used it for small molecules, and then it's actually been very useful for large molecules. So, for example, Alnylam is a local company in the Boston area, and I was asked to be on the scientific advisory board from the beginning, and Alnylam got the very first, I helped them, and they got the very first what's called siRNA approval, and basically they did it by putting it in nanospheres. That's for a very rare disease called ATTR amyloidosis. That's been very exciting. And then in 2010, I myself and one or two other people, we started this company just actually less than a block from myself, which is called Moderna. And the whole idea of Moderna was to develop new drugs of messenger RNA. And messenger RNA can be very, very useful. But again, if you try to inject it into the body, it's destroyed right away. So what we do is we put it in these little nanoparticles, and they, in this case, made of lipids, and basically, but they still have this coating, and basically we inject them in, and they last long enough to make different things. Probably the classic example that many of you have heard about is the vaccine for COVID, and so basically we've made a messenger RNA, put it in nanoparticles to make a COVID vaccine, and hopefully that will be approved by regulatory authorities in on the 11th. At the end of this week. So at any rate, all these drug delivery things actually enable many therapies that would never have existed before. By the way, every company, Pfizer, BioNTech, Moderna, CureVac, Novavax, all these companies use nanoparticles to deliver like we originally designed. They've improved them from what we did, but to deliver and protect mRNA. Now, I want to go on and tell you some other things that we created. One of the things that I became curious about when I worked in the lab at the hospital was how did materials find their way into medicine? And when I looked into this, I found it was almost always done by medical doctors. And what they do is they'd go to their house and they'd find some object that kind of resembled the organ or tissue they wanted to fix. So it turns out that the artificial heart is a lady's girdle material, so it has a good flex life. And a mattress stuffing is actually used for breast implants because it's squishy. So I thought, well, we could do better. Rather than take materials from your house, what we could do is actually use what I'll call chemical engineering design. Ask the question, what do we really want from a biomaterial, from an engineering standpoint, chemistry standpoint, biology standpoint, and then design it from scratch. So we picked an example. And the example I chose was synthetic degradable polymers. The next slide shows an example of how almost all degradable polymers dissolve. And they dissolve by a process that I'll call bulk erosion. So if you start on the top, it looks like a sheet, but then it gets spongy. And then by the time you get to the bottom, it falls apart. That may be okay for some drugs, but for others, it's as you see, it gets so porous, the drugs could just burst out. So if you had a cancer drug or if you had insulin, that would be very bad. So we said, from an engineering standpoint, we don't want the polymers to dissolve like that. We want them to dissolve like as shown in the next slide. Surface erosion. And we wrote on the blackboard all the criteria that we wanted to do this. So we wanted to make, you can make choices like what should cause the polymer to dissolve, like water or an enzyme. Well, everybody has different enzyme levels, but you all have excess water. So we said, we'll build into the new polymer the fact that water's got to make it degrade. And then we said, if we do that, what should the building blocks be? We need them to be safe and non-toxic. And what should the bonds be? So what we came up with, without going into a huge amount of detail, is shown in the next slide. And it's a polymer it's called a polyanhydride, and there are two parts to it. It's what's called a copolymer. It's a basic acid and carboxypropane, carboxyphenoxypropane, and it's connected by anhydride bonds. But our theory was that if you could change the ratio of these two, you can get almost any erosion rate you want. And that turns out to be true. That's shown on the next slide. And even if you take a thin system, you can make this, here we're on this slide, we're changing the ratio of the two units. 79% of the one, 55% of it, 15%. But if you go to 0%, you can imagine that that'll take about 4 years to dissolve, whereas the 79% one is gone in 2 weeks, and we could make it go faster. So one of the things that's been very important to me, I love doing the basic research. I love doing curiosity-driven research, but I also want to see this help people. So in 1985, a young neurosurgeon named Henry Brem came to see me. And he was then just starting as a neurosurgeon. Now he's actually head of neurosurgery at Johns Hopkins. But he wanted to figure out a better way to treat brain cancer. And the worst and most common form is shown in the next slide, glioblastoma multiforme. And no matter what you do, the mean lifespan is less than a year. And the only drug that was approved at that time for use is shown in the next slide. And this drug is incredibly toxic. You give an injection, and it causes terrible side effects to the liver, kidney, and spleen. So Henry and I had an idea that maybe we could come up with a new paradigm. And that idea is what we'll call local chemotherapy. And this is shown on the next slide, where what the neurosurgeon might do, like Henry, is line the surgical cavity with a polymer that contains BCNU, the drug. Now, normally it would only last 12 minutes, but when it's in the polymer, it's protected from degradation, and it will last as long as the polymer lasts. And we could make this polymer last any length of time, like a month is what Dr. Brem wanted. So we did that by changing the copolymer ratio. But the power of this is you get high concentrations in the brain where you want it, and low concentrations, this is the fourth bullet, in the brain where you want it to kill the tumor. So high concentrations in the brain where you want it, but low concentrations everywhere else. So when we first proposed this, you have to raise money. And the way we would raise money is by writing grants to the government. And they'd have what are called study sections. These are other professors who review what we do, and they decide whether we should get the money. And I was very excited about this, but our grants did terribly. The next slide gives a brief summary of what happened. In 1981, when we first proposed this, the reviewer said the polymers can't be synthesized. But I had a really good graduate student, Howie Rosen, who later became CEO of a $12 billion company in drug delivery, and he showed they could be synthesized. Then people said, well, okay, but these polymers, they'll react with whatever drug you put in because of these anhydride bonds. But another postdoc, Cam Leung, and he showed, now he's a very distinguished professor of a chair at Columbia University, he showed that didn't happen. So then we sent it back again, and the reviewer said, well, we still shouldn't fund this because these materials are fragile, they'll break in the body, they're low molecular weight. But I had another couple of postdocs, Edith Mathiowicz, who's a full professor now at Brown University, and Avi Dome, who's dean of pharmacy at Hebrew University, and they showed that we could make really strong, high molecular weight polymers. We sent it back again, and the reviewer said, well, we still shouldn't fund this because this will be a new polymer, it's going to be toxic. But Cato Lorenzen, one of my students, who later became the dean of medicine at University of Connecticut, and also received the National Medal of Technology from President Obama for this work. He showed that, and other work showed that it wasn't toxic at all. So then the reviewer said, well, okay, but the drug won't diffuse far enough to kill the remaining tumor. But I had another graduate student, Mark Saltzman, who Yale University asked to be their initial head of biomedical engineering. And he's won all kinds of honors. And he showed it did diffuse far enough. Anyhow, this went on and on until 1996 when the FDA approved it. It was the first time in over 20 years the FDA approved a new treatment for brain cancer, and the first time they ever approved this idea of local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well all the MIT people did. They got great jobs and honors, whereas the reviewers, they haven't done so well. Now let me just show you a little bit about how this works. I'll show you, again, a picture of a brain with a little wafer in it on the next slide. And on the next slide, we'll show you, the following slide, I'll show you how it usually looks. They put several of these in before closing up the brain. Next slide. Next slide. I don't like to leave those on too long. So basically, there have been a number of clinical trials. This was one of the very first ones. And it's not a cure by any means, but it did show that you could prolong life by, in this case, in this study, about 5-fold increase in survival compared to controls, which are conventional best treatments. And even a couple years ago, they analyzed, the next slide, about 60 clinical trials, again, showing that it was statistically significant. Actually, so I should have pointed out that it got approved in 1996. It's still used today. Basically, it's been approved in over 30 countries and used on thousands and thousands of patients for now the last 24 years in these 30 countries. It also created a new paradigm for other treatments, in particular, and this, what I'm going to show you shows how you never know where things will lead. So this is treating cancer. But one of the biggest problems in heart disease, as you probably all know, is you get blockage of blood vessels. So a way that people treat that is what are called by putting stents in. These are like Chinese finger puzzles to prop the blood vessels open. And a picture of that shown on the next slide. But what happens is, as you may know, is about 60% of the time, you get the smooth muscle cells proliferating when you put these stents in, and the blood vessels close, and the patient can die. And so, and you have to put a new stent in the best case, but death in the worst case. So what they now do is several companies like Boston Scientific and others, they make these coatings of polymer, and they put a different anti-cancer drug in, even though it's for heart disease. That drug is Taxol, but it's anti-proliferative, and it locally delivers the drug. If you gave it systemically, patient would die. But they give it locally, and it's like 1/1000th the dose, and it keeps the blood vessels open, and millions of these are used every year. So these are two things that I wanted to go over with you on drug delivery, but I want to now turn and end the talk on how you maybe could use materials to someday create new tissues and organs. And here I've worked very closely with Jay Vacanti, who's a good friend of mine and a surgeon, initially at Boston Children's and now at Mass General. And about 30, almost 40 years ago, he came to see me when he was heading up the liver transplant program at Boston Children's Hospital. He wanted to make a new liver for this little baby on the next slide. And the problem is that child has to get a transplant. If he doesn't, he would die. And so Jay once asked me, he said, well, Bob, could we make new tissues and organs from scratch? So our idea, this is from a paper we wrote in Science, next slide, that's been cited, I think, almost 8,000 times. Next slide. Our idea is we could take almost any cell type, and these are shown on the left. By the way, we could also take stem cells, but people didn't talk about stem cells then. So here we put different cell types in, we dissociate them. If you injected these cells at random, not much would happen. But the cells are smart, and people have shown that if you put cells close enough together, they can recreate tissue structures. And so here what you see is an example of a polymer scaffold that we want to make where we could put the cells close enough together. Then we grow it in a bioreactor in vitro. And then the idea is you could put it in the patient and make virtually any tissue you want. And once again, we created new materials. The next slide shows some examples of that. We either use conventional FDA-approved materials like lactic glycolic acid, and these are degradable, but sometimes cells won't adhere to this. So what we wanted to do was build in the ability for cells to adhere if we wanted to. So we also created new polymers like lactic acid co-lysine. And notice on the bottom picture depiction, you see where the lysine is the NH2 group. So for the chemists in the audience, you could use what's called a carbodiamide reaction and link almost any set of amino acids or other things to it. And that's actually helped us in many different tissues we've tried to create. Now, on the next slide, what we've done is we've formed them into fibrous structures. And here, like you see, there's a lot of porosity, and this we put liver cells on. But the beauty of engineering is you can make almost anything. So Prasad Shastri, who was one of my postdocs, he now runs an institute in Germany, actually had this idea of using CAD-CAM techniques. That's computer-aided design. And then you follow it by something like three-dimensional printing or different foaming techniques, and you could make these systems in any shape at almost any porosity. So as an example, he picked a nose. And what I'm going to tell you now is speculation, but let's say 30 or 40 years from now, somebody goes to a plastic surgeon and they don't like their nose. They want a new nose. So our idea was that you would have a computer screen. You could pick your choice of 40 noses. So the next slide shows an example of that, where he's on the left, you see a nose of that doesn't have cells on it, but it's 98% porous. And then you could take the cartilage cells actually from the patient themselves. I'll show you an example of that in a few minutes. And you could make a nose. Now, this is like a regular nose, but if you wanted, say, an upturned nose, you could do that. You could just take a little bit of the cartilage off the spongy one on the left, and maybe somebody would want a hook nose. They probably wouldn't, but if they did, we could make that too. So just to show you a few examples of making cartilage, next slide. Why don't I show the next slide? So with the Vacantes and Lisa Freed and Gordana Vunyak, we actually took nude mice and took their own cells and made, as you can see on the bottom, we redid his skull. And on the next slide, we redid the top animal's cheek. And if you open them up and look at them, it's pure white glistening cartilage. And I'll show you that on the next slide. And if you actually do what's called histology, that's a way to judge about what you made. What you'll see on the next slide is it looks like almost a textbook of cartilage. The characteristics of cartilage are having glycosaminoglycans, of having type 2 collagen, of staining with saffron and O, and having these chondrocytes in characteristic lacunae, which you see on the bottom two parts, C and D. And finally, staining for chondroitin sulfate. That's very characteristic of cartilage, and that's with an Alcian blue stain. So it looks just like cartilage. The only thing is it's not as good as regular cartilage. If any of you are runners and thinking we can help you, we're not there yet, but we can help people cosmetically. And the United States Army came to see Jay, myself, and one of my former postdocs, Linda Griffith, a few years ago, and they said, we have people coming back from soldiers, coming back from Iraq and Afghanistan, said, could you ever make new body parts for them, like a new ear? So on the next slide, what Linda did is she made an ear, as you see on the top, but if you look on the bottom right, what you see is a high-powered scanning electron micrograph of polymer fibers where we've taken chondrocytes and they're growing on the scaffold, and over time you'll get pure cartilage, and the scaffolds will disappear because they're degradable. In fact, they'll break down into water and carbon dioxide. And what that means is when you put this ear with the human cells, you could put it on a rabbit. And this was actually done by the Vacantes. The next slide shows a picture of the human ear on the rabbit. And you can see that. And finally, we go to the next slide because we actually did do this on patients. Next slide shows a 12-year-old boy that my colleague Jay treated. And this 12-year-old boy, like others, likes to play baseball. But if he ever got hit in the chest with a ball, he could die because he's got no chest covering his heart. So Jay operated on him. We made a scaffold for him. And Jay took the boy's own cells and made him a new chest. And he's now 6 feet tall and doing well. Another thing we did is skin. Next slide. In this case, we actually licensed this to some companies. And here's a little boy who's very badly burned. And you can see that. Now here the product is going to be neonatal skin fibroblasts. Next slide. And you can actually cryopreserve these. But then what you can do is put them on the patient, on the little boy, at the time of the injury. Next slide. And let's come back 3 weeks later. If we could go to the next slide. Yeah, thank you. So you see he's doing better 3 weeks. Now let's go to the next slide and we'll see him at 6 months. And as you can see, he's doing much, much better. Now these have been approved for patients with diabetic skin ulcers and also patients who are burned like this little boy. And they're now used by several companies and they've treated many, many patients. I thought I'd do one last example to illustrate a slightly different point. Not only can you make new tissues and organs, but you can, and we're working on many different kinds. We're working using these principles on making new blood vessels. That's now just being done and completing phase 3 clinical trials, final clinical trials. We're working on spinal cord repair to hopefully help people walk again who are paralyzed. We're working on hearing loss, in that case, actually by discovering some new molecules that may stimulate hair cells. But and we're also working on making a new pancreas for diabetics. But what I thought I'd end on is one other thing, which is that you could also use these approaches, and we are, and some of my students are, as well as other people, make organs and tissues on a chip. So here I want to tell you about the work of several of my students when they were in our lab, and they've now actually started a company to do this. So let's say you wanted to make a heart on a chip, and the next slide shows what they did. You can take what are called cardiomyocytes, and if you give them the right media and put them on the right polymer scaffold, they will have almost the exact architecture of heart tissue. You see sarcomers and an intercalated disc. And what's really striking is you can take these systems, and if you look at them under a microscope, you could take 6 million cells. And I will just show you a video on the next slide of this. And we're not shaking this or anything. But what's really amazing is here you have 6 million cells and they're beating like once. It's like this muscle, your heart beating, and yet it's in a dish. It's not in an animal, not in a person. So you have these 6 million cells in a dish, and that's what it looks like. So what you can actually do, and I'll go to the last slide that I want to show you today, is actually take an EKG of it. So you can really, these actually really mimic exactly what happens today. So now what several of our students, Milika Radzek, who's at the University of Toronto, and Gordana Vunyak, who's a chaired professor at Columbia, they started this company, which they asked me to help a little bit on, called Tara Biosystems. And because you have this heart in a chip, what they're doing is you can screen for toxicity and see if the heart tissue, if the molecules that people are studying cause toxicity. You could also actually use it to test new drugs. Are they effective in taking heart tissue on this heart on a chip? And not only can you make a heart on a chip, others have made livers on a chip. And another one of my former students, postdocs, Gio Traverso, he's made a gastrointestinal tract on a chip. And another ones have made lungs on a chip. So this has the potential to someday minimize animal testing, and I hope minimize human testing. So, but you can make all these. Finally, it's interesting. People have done things with this tissue engineering that I'd never dreamed of. Like one company I know of is making leather, and quite a few companies, including one by Shulamit Levenberg, another one of my postdocs, they're actually making meat this way, because you could actually engineer meat by, again, using the right kind of cells, the right kind of media. So it'll be interesting to see where this all goes. But already what's been wonderful is that you can use it to make some new tissues and organs to really help people who are burned and with other medical problems, and you can develop these organs and tissues on a chip. More broadly speaking, as I close this talk, I want to thank the organizers for asking me to do this. And what I have tried to get across is how chemistry and engineering can do things that I hope are having a big impact on medicine, everything from new drug delivery systems to COVID vaccines to creating new tissues and organs. As you could see from what I went over, that I had certainly a very rocky road for early in my career, where people were very skeptical of these ideas. I think if I have any things to impart to the people in the audience, not that I'm any expert, but it's to dream big dreams that you hope can have a big impact on science and medicine, and recognize that you may often have a lot of obstacles, people telling you that your ideas will never work. And I think it's really important to not give up, to keep pushing and pushing, and hopefully your big ideas and big dreams will come true. Thank you so much again for having me. It's been an honor to present this.
Thank you, Bob, for a very fascinating and inspiring lecture. We have a number of questions that have come in, and I'm going to quickly ask you a series of questions one by one. First is, let me start with one of the questions that came in. I'm going to introduce that question with my own personal experience with you over the last more than 25 years, almost 30 years. One of the most impressive things about your work and the way you manage it is time management. Even 2 days ago when we talked, I sent you an email, and within a few minutes we were on the phone together, and it was only early morning in Boston, on the east coast of the US in Florida. So the question was, how do you balance between, how do you manage your time given your multitude of interests? And also, how do you manage between faculty research work that you do, mentoring students that you do on a global scale, and also your commercial research and involvement in FDA and boards of companies?
Well, that's a very good question. I've been asked that before. And actually in, I think 2009, Nature wrote a 3-page article just where Helen Pearson, one of the editors, followed me around for the entire day. And it was educational even for me. I think, well, what I do is, well, first I have lots of short meetings, 15-minute meetings, with different people. And, and I also have I'm very lucky. I have a terrific staff at MIT, both in terms of people in the office and people in the lab, and they help me out a lot. And, and actually the other thing I've done, which probably will sound crazy, but I don't, it's travel. Like I never stay any place very long. I always try to get back as quick as possible. The example I gave, which is true, is 5 years in a row, I had to go to Israel for something like a lecture or honorary degree or something. And each time I actually didn't use a hotel. I flew from Boston to New York to Tel Aviv. I gave the lecture. I had dinner with some of my students, and and then they have a 1 a.m. flight, and I just flew right back. And so I really don't, spend a lot of time schmoozing, I suppose. Even when I was in Singapore last, I did pretty much the same thing. I was in Singapore for just about 24 hours, and I was able to give a lecture and have a number of meetings, have dinner with a bunch of my students, and then hop on a plane. And it was a long ride back, but but those are some of the things I do. Commercially, we maybe get involved in a company in a year. Pretty much every company I've been involved, actually, it's my students that are also involved, and I do a lot, and I have a very close relationship with them, and I it's like I work through them, and I I'm often on a board of directors and scientific advisory board.
Thank you, Bob. One of the questions that came from one of the young scientists is recently there have been reports of, at least the question, that's what it asks, apparently increasing cases of allergic reactions to mRNA vaccines as a consequence of polyethylene glycol. Do you have any perspectives on how to address it in the future, or is it an issue?
Yeah, well, first, I think it's speculation. There have been, I think, a few very rare incidences of allergic reactions to the vaccines, which could be for many reasons. I think there's speculation that it's polyethylene glycol. Whether that's true or not, I don't know. But there are some. But the amount of we people may not realize it, but we take lots of polyethylene glycol every day. I mean, it's actually in different things. Like, for example, if anybody's constipated, which lots of people are, you take something called Miralax, and that you take a tablespoon of it a day, and that's probably 1,000 times more than what's ever in these little nanoparticles. So I don't know that that's true, but how you address it is important. I mean, you have to do studies to try to understand what causes it, and it could be any number of things. It could be to impurities in any of the preparations, whether it's polyethylene glycol or other things. So I think you have to step by step take that apart. But so far, it's important to realize that they're very, very rare reactions. It's happened a couple times.
Bob, one other aspect, perhaps a unique and a very impressive aspect of your work, is that you have remained a faculty member and an active scientist throughout your career without leaving academia. But at the same time, through startups and also through established major companies, you have found a way to have significant impact on industry and society, especially at the intersection of engineering and medicine. So especially for the young people in the audience, do you have some suggestions on how to balance, on the one hand, basic academic research with practical and applied research, especially in the area of medicine, and also basic research in engineering becoming very applied and useful product in biomedicine?
Yeah, well, I think my advice, and actually what I did is I always put the MIT work and the basic research first. That's always been my top priority, working with the students working in the lab. But one of the things that became important to me, you work on things like some of the drug delivery work I talked about. I remember I worked on for 10 years. We published papers in Nature and Science and other places, and sadly, nobody was using it. And I realized, and that includes even the things that would lead up to the nanoparticles that are, being used to, to treat COVID vaccines today. But I realized if I didn't take the initiative of trying to move these things forward, practically, nobody else would. So, I mean, maybe I shouldn't say nobody else would. Maybe over 50 years somebody would, but it doesn't happen easily. So, to me, it was important to not only do the basic research, but once that basic research was working and could be useful, it was very important to me to see it help people. And if I didn't get involved in companies, probably it wouldn't have, or it certainly would have happened a lot less.
One of the people online asked the following question. So, can you comment on the potential of bioprinting technologies? How far away are they from clinical use?
Yeah, well, I think bioprinting is very exciting, but I also think that the number one issue in terms of there's a couple of things you can use it for that are important, right? One is like an organ or a tissue on a chip, and I think that's exciting, and that could probably, that could be a surrogate for, animals and humans at some point. I think you'll still need to use animals or humans, but you could do high throughput screening. So that, I think, is already there in some cases. There are companies doing that, and I think it has accelerating, some drug development, some drug toxicity. So I think that can already be done. I think in terms of really making tissues for patients, that's going to be longer. And the reason for that is understanding the biology well enough. I think we really need to understand the biology to guide what you want to 3D print. And I think that will take longer, but I think someday that'll happen as well.
Yeah. Several people asked a question. I'm going to combine all of them into the following question. We have time for only one or two more questions. So at the start of your career, like most scientists, when you are young and trying to prove an unpopular or unconventional idea, you faced a lot of skepticism from established researchers. Do you have particular advice for our young scientists who are joining us online on what kept you going? And what advice do you have that now this is almost 45 years later, the world has changed a little bit. Should they do something differently from what you did then?
Well, I think that, there's a couple of things. I think the most important thing, I think, for young scientists is to do just great research, to dream big about really asking really big questions that, if you're successful, might have a really big impact. I think you should recognize, though, that if you do that, sometimes people are gonna, you might run into what I'll call conventional wisdom, and people won't like it if you upset the apple cart, so to speak. I'm sure you've seen that too, Subra, and in your work. And so I think when you do those kinds of things, I think you just have to hang in there. You have to believe in yourself, and you have to keep trying. And it may take a long time, but I always believe in dreaming big and not giving up. And I guess I've been very stubborn, and I think it's just important to ask big questions and then not give up easily.
Thank you. And Bob, I know for several decades how busy you were traveling. So here is a personal question to you. Have you missed travel in the last 8 to 12, 10 months or so?
I miss, it's funny, I don't miss being on the airplanes. One of the nicest things for me traveling, like the last time I was in Singapore, I had dinner with probably about 15 of my students, and that was great. And when I mentioned Israel, I think I was there and I had dinner with about 20 of my students. So seeing people that you care about, in different places, I miss that. But I have to say, not being on an airplane, I don't miss that. And it's interesting. I look back at my life, and it's now I'm 72. And last year, 2020, is the only year I can think of where I have not been the least bit sick. I didn't get a cold. I didn't get anything. And I'm sure it's because I didn't travel. So, so I've gotten very, very used to Zoom, and, probably on Zoom 12 hours a day. So I think the answer really to your question is it depends. Some aspects I miss, a lot of aspects I don't miss.
Well, thank you, Bob. I wish you many, many more years of good health and free of any even mild flu, so that not only for you, but also for the global scientific community. And on behalf of NRF and the organizers and all the people joining us online, let me thank you for taking time late in the evening, your time, to join us online and giving a very inspiring lecture. So it's also a great pleasure for me to see you and connect with you in this particular way. So thank you very much.
Well, it's an honor for me. It's wonderful to see you again, and it's wonderful to be able to speak to all the young scientists in the audience. Thank you so much for having me, and thank you, Subra, very, very much.
Thank you. Bye-bye.